Okay, Dr. Gevorg Grigoryan is co-founder and chief technology officer at Generate Biomedicines, where he oversees the Generate platform that enables the generation of new drugs on demand across a range of biologic modalities. In addition to his role at Generate, he is a research associate professor of computer science, biological sciences, and chemistry at Dartmouth College, where he obtained tenure in 2017. Gevorg's research focuses on understanding the relationship between amino acid sequence and structure, seeking rigorous ways of inferring the principles governing protein folding, interaction, and function from data. Gevorg holds a bachelor's degrees in computer science and biochemistry and his PhD from MIT and postdoctoral work at the University of Pennsylvania. Gevorg has authored many peer-reviewed manuscripts at top journals, and his work has received awards and recognition from the Sloan Foundation, NIH, NSF, and ACS. And with that, I hand it over to you.
Sorry that had to be so long. I didn't realize they were going to read everything when they asked for the bio. But thank you. Oh, okay, good. Well, thank you so much. Thanks for the invitation. It's been a fantastic meeting so far. I want to talk to you about programmable generative protein design. Now, I've been in protein design for a couple of decades now, for a while, and I can tell you that there's something really special happening in the field right now, where machine learning methods are fundamentally changing our ability to generate novel, potent, functional protein matter. And in fact, I would say in the space of generating molecules in general, there's something really special going on. You can even just tell from the lineup of speakers here. Andrew, what an amazing talk. I mean, wow. But also Rafa and Posu coming up very much in a similar arena. So I'll tell you a little bit about what we're doing to advance this field at Generate, but also because this is such a special moment in the field, I'll try to tell you a little bit about what has been hard so far, what is happening right now that's changing that, and what might we be able to expect next. So first, I hope I don't have to convince this audience that being able to design proteins is a useful pursuit. They do all sorts of really interesting things in nature, from chemistry to all sorts of energy transformations. And we routinely think of them as potentially being best-in-class solutions for other complex problems like complex disease and all sorts of nanotechnological applications. So long as we can actually gain mastery of this molecular class, which of course has been really difficult. Nature, of course, has been at it for a while and has been quite successful. But I would argue that even for nature, it hasn't been particularly easy. While it has designed or evolved lots of different functional proteins, it's taken it billions of years, which doesn't seem exactly efficient. And also, most functional proteins we know today actually evolved a long time ago. And since then, nature's really focused on editing them a little bit here and there for improving functionality. When we as humans have tried to engineer proteins, we too have had the most luck with starting with natural examples and then modifying them to kind of tune the function here and there. But the point is, whether we're doing it or whether nature is doing it, this has been hard. And one kind of very high level way to think about why is that protein design is really a complex constraint satisfaction problem, which is to say that before you're even allowed to think whether your protein does anything, much less the thing that you want it to do, it actually has to be a protein. It has to be a legal member of this class. And in order to do that, it has to meet all sorts of constraints. Like proteins have to be nicely packed. They don't like to have voids. They can be overpacked, but that doesn't work either. Everything has to be zipped up with these atomistically precise hydrogen bond and other types of interactions. And all of this has to spontaneously come together using an alphabet of only 20 amino acids. So this has just been difficult. So the question is, and all of this is, even before you worry about the function, which is also very difficult, but if the protein isn't a member of this class, then of course you can't even think about function. So the question we asked at Generate, we live in an age of machine learning doing some amazing things. What can we do in order to see some of the benefits of ML applied to proteins? After all, we've seen other examples in other domains where ML has done amazing things at being able to satisfy very detailed, very precise constraints, while also having kind of overall semantic consistency within the generated objects, like in images and other examples. So how can we do something similar in proteins. Well, one of the things that helped a lot with images, there were many advancements, of course, but one important one was diffusion models. And you can think of diffusion models as solving constraint satisfaction by breaking that concept into lots of small steps. It turns out that if you train a neural network to undo the impact of progressively added noise onto your sample, all the way to the extreme where all you have is noise and the impact of the sample itself has been completely lost. Turns out if you do that, then you can effectively reverse the process and start with something that's actually trivial to generate, which is just a random noise sample, and ask the model to denoise it progressively in such a way that it now yields a sample that would have led to that noise pattern had you run it in forward. Now this, for many of you, this is trivial, and sorry for over-explaining. And then for some of you, this will probably sound like magic. And so if you're looking for some amount of intuition as to why this even works, think about if someone asked you to describe the distribution of all possible houses. Right? You know exactly what a house is. You can know a house when you see it. But how would you go about describing all possible houses, that probability distribution? That seems like very hard. What might be easier is for you to describe the process by which a house is constructed from kind of elementary building blocks. Any builder knows how to do that. And so if you describe that process in a general way, you would then say, well, anything that emerges from that process is by definition a house. So there you've kind of implicitly parameterized the space of all possible houses. And you've also furnished yourself an ability to draw from that distribution. And diffusion models do something similar, but in a very much a generalized manner. So at Generate, we built, we've been toying around with generative models of proteins for quite some time. And we built a diffusion model for proteins that we recently published. It's called Chroma. And we thought a lot about what are the kind of key features that a diffusion model or any generative model of proteins ought to have in order to actually have practical impact on real applications. And I'll very quickly kind of mention just 3 of these aspects. One is scalability. So it turns out if you actually do what I said earlier and take kind of standard diffusion architectures and standard thinking about molecular ML and apply it to proteins, you'll get a system that scales either quadratically or cubically with the size of the protein. And that's because basically all parts of the protein have to talk to each other, and that's where that at least quadratic scaling comes from. Now this is a well-known problem in molecular simulation and computational physics, and it's never the solution, right? Whether it's in astronomical simulations or molecular simulations, the answer is always look for more scalable approaches, and we did that for proteins, and I won't go into the details, but the fact is that we're able to achieve essentially linear-like or just linear scaling while still allowing all parts of the system to talk to each other. I don't have time to go into the kind of the technical details. They're all in our preprint that was recently put out. But the punchline is that you can do this. You can start with random kind of nonsensical sets of chains of proteins, and very cheaply and very quickly generate what look to be completely realistic, completely plausible proteins that could have just come out of the PDB, but in fact they didn't. And they don't relate to anything that's in the PDB, and they don't seem to, have sequences that seem to have evolved from anything in the PDB. I have to say that when this first happened at Generate, this was, some time ago. For me personally, as I mentioned, I've been in this field for a while. This was a bit of an outer body experience because I knew how much work and energy would have gone into creating just one of those objects that simply satisfy all those constraints that I mentioned before. And is novel, is not something already in the PDB. This could have easily taken months, if not years of PhD time. And now we had a thing that you can just push a button and it would generate these IID, as quickly as you want, as many as you want. So for the next 2-3 slides, I just wanted to show you how some of these things look like because I find them just fascinating. So these are all different structures generated by Chroma. You can look at single chain structures. Again, they look like something that could have potentially come out of the PDB, but didn't. You can easily generate multi-chain structures, as many chains as you want. In principle, this scales very little. Here's a 2,000 residue complex. We've generated 4 or even larger, 4,000 or even larger complexes. Again, this just costs a minute or a couple of minutes of GPU, commodity GPU time. So it's not, you can do it on your laptop. So scalability is the first important thing. The second, access to likelihood. So not only we think should you be able to generate samples, but you should be able to evaluate how likely your model thinks that that sample is. Again, I won't be able to go into the technical details, but you can think of it as something that's very akin to essentially free energy or probability from statistical mechanics. If you want to do any kind of quantitative analysis, you need to be able to have access to that object from your model, and Chroma is able to produce that. And finally, and perhaps most importantly, is programmability. It's wonderful for me, again, this was just amazing to be able to go from not being able to generate protein samples to being able to generate it with ease, but ultimately, who cares? These aren't supposed to do anything special, and if they do, we don't know what they do. All right, so really the important thing is to be able to drive the generative process towards proteins with properties and functions that you actually care about. And that's something that's inherently baked into Chroma. So it turns out what the foundational model, kind of what the underlying model really learns is something akin to the probability distribution that any given set of coordinates, any given protein could have come out of the PDB. And it turns out that as long as you have a model which you can take some proposed protein and then evaluate how likely it is to have the property that you're looking to produce, as long as you have such a model, and these are called classifiers, then you can staple these two objects together, this unconditional probability distribution that the model has learned, and a conditional probability that your classifier is producing. You can staple them together mathematically correctly to drive a conditional generative process. So now when you push the button, you don't just get random proteins, you get proteins that are conditioned on having some property Y, whatever that property is. Again, as long as you have your classifier as appropriate. And so we've been playing, having a lot of fun with this concept. In our preprint, we built a couple of different classifiers to enable conditioning. Turns out it's actually pretty easy to condition on symmetry. I'll show you some examples. Conditioning on substructure seems to be important. This is for grafting functional motifs or transplanting scaffolds, etc. Conditioning on arbitrary 3D shape, being able to say what shape you want your protein to have. And even some more far out kind of crazier types of conditioning, like conditioning on class, but also just conditioning on pure text, saying, just type what you want your protein to be or to do, and have your model be able to actually generate a protein to do this. This last one, as we mentioned many times in our preprint, we did this kind of for fun. We're not proposing that this is actually a useful model at this point. It's not very good. It can only be conditioned on essentially the kinds of things that are captions in the PDB, and has learned that mapping. But this we did kind of more just to, to show what's going to be able to be possible, we think, in relatively short order. Okay, so again, some more examples. Conditioning on symmetry, just as easily as you can produce single chain samples with no additional cost, you can actually produce samples of any given point symmetry group. Here are more symmetries. These could be either closed symmetries or fibers, and whatever you want. You can condition, like I mentioned, on some structures. You can take catalytic domains and embed them into otherwise IID sampled structures, which is just sort of a crazy concept that I think was just never possible before. You can do things that seem completely useless, like build proteins that are in the shape of English letters and numbers, or you can come up with slightly more useful versions of that kind of capability. And as I mentioned, conditioning on more complex things like cath topology in various classes, as well as just straight up text. I mean, just to kind of show you this example, here we're saying, conditioning on the tag crystal structure or Rassmann fold, it produces this. This is a canonical Rassmann fold. You can judge how similar it is. Again, we're not saying this is very good yet because it's trained on very limited data, but you can start seeing the point or crystal structure of a fab. To me, this is a pretty fab looking structure. This is a real fab. So, it will be useful. It perhaps is a toy model at this point. And then, of course, we've computationally validated all this. You can show examples of these things, even the crazy things that are actually refolding from a completely orthogonal set of structure prediction models, those that we didn't build, like AlphaFold, RosettaFold, OmegaFold, et cetera. You can find refolding examples, which of course is not proof, but it starts showing that these are all plausible solutions. And of course, we have done experimental validation as well. I don't have time to go through these today. And the preprint doesn't have experimental validation. We're hoping to be able to share that and more, thank you, when we're able to publish a peer-reviewed article. But just to kind of summarize at a high level what we did experimentally, maybe just to take a step back, protein design has been around for a while. Some really impressive things have been designed. Enzymes, binders, assemblies, you name it. So just being able to do it is no longer impressive. What hasn't been done is to say, here's a model, it's a black box, you can push a button, out comes a protein, I'll push it a bunch of times, and I'll make every protein that the model proposed exactly how it proposed. I'm not gonna put any filters, I'm not gonna have experts opine on whether this is likely to work or not. If it looks stupid, we'll make the stupid thing. If it looks smart, we'll make the smart thing. And do this, let's say 100 times or however many times, so that we can have an unbiased estimate of how much of what the model learned is real. And that is exactly what we did. In this case, we pushed it about 300 times. We made all the proteins exactly the way the model proposed, either unconditionally or conditioned on something. We didn't filter, we didn't employ other tools, we didn't opine on the energetics, we didn't obsess about the clashes. None of that happened. And so in that light, the results are a lot more informative, in my view, than a typical result of experimental validation from design study. And so again, I'm not going to dive into the details, but by and large, what we saw is that these proteins express, they have biophysical properties that are consistent with what the model suggests that they should be, and they have structures that are atomistically superimposable onto the proposed, or onto the model structures. So, why this is different, I think I've covered this in the previous slide. This is kind of singular model. It's able to generate across different classes. You don't have to filter it. It has a stable, non-trivial hit rate, and it's sort of is able to access programmability. Maybe I'll conclude with that. I think what I started this talk with is that it's been hard to do constraint satisfaction, and that's really hampered the field to even start thinking about function. Well, it really seems like, in our work and other work that's happening in the field, it seems like that's problem, if not solved, it's just about to be solved. Which means that the emphasis will be shifted towards function, building models of function, building conditioners of function so that we're able to produce more potent, more interesting proteins, which is of course going to shift the emphasis towards data generation. And that's a lot of what we talked about from earlier talks, or a lot of what we heard about from earlier talks about from Recursion and Citro and others, data generation to produce better conditioners and better functional models. A lot of people to thank. There's a big team at Generate that's responsible for this, both computational folks and experimental folks. I'll just point out specifically John Ingram, who's the head of machine learning at Generate. In many ways, this project was his brainchild, and it's been phenomenal to work with this team. Thank you, and I'll take any questions if I have time.
Great presentation. So I have a question, because the generative AI in protein design is super hard. That there are also other biotechs that doing things like that. For example, like Profluent, they published like one paper in Nature Biotechnology. It's also using generative AI in protein design, and their focus is on the sequence part, protein sequence, and also other biotech, which is also one Chinese biotech, which called biogeometry. Their focus is on the structure of the protein. So I was wondering, for Generate Bioscience, so you're focusing on the sequence of the protein, of you focuses on the structure of the protein, of you consider both parts of the protein?
Oh yeah, very good question. So what I didn't have time to specify is that there's a structure layer, a sequence layer, and a side chain layer. So when you generate these proteins, they come with a structure, sequence, and kind of a side chain representation, and you can condition on any of those layers. But that's not, so the other point is you're absolutely right. Lots of people are active in this field, and I think lots more will be. I mean, this is really just the beginning. We'll see lots of different models. We might hear about not unrelated models later today. So yeah, very, very active space, very kind of exciting time for the field.
And another question would be, so for example, you generate like millions of proteins, and how you build like high performance lab techniques to verify these proteins. You can't just generate them then leave them alone, right?
Yeah, absolutely. I mean, you're making a very good point, which is also something I tried to make at the end, which is now that this is possible, now that you can cheaply take credible shots on goal, it will all become about the scale at which you can validate, the scale at which you can measure, tune, build conditioners. So yeah, I couldn't agree more with your statement.
That was lovely.
Thank you.
You took like 300 shots at the end. You expressed them.
Yeah.
Was there a correlation between what was refoldable by AlphaFold and what was expressible by E. coli?
So there wasn't, which was kind of disappointing and unexpected, because there were other kind of papers in the field that proposed that there might be. So I mean, correlations of course are funny because they depend on the reference distribution you're sampling from. But from what Chroma is sampling, the refolding rate does not seem to be, or at least is not as strong. I mean, from within the number of samples that we sampled, we didn't see a correlation with success.
Super cool stuff. You know, clearly, like we've seen diffusion work very well for structure-based protein generation. But I want to talk about a class of proteins like they're disordered, right? Like intrinsically disordered proteins. They're valuable proteins. Even though they don't fold well, express well, they're still viable proteins. So how are you going to design using these types of models? How will you design to them as binders, enzymes? How are you going to do that?
I mean, this is very valid critique. So by construction, right, this model learned from just the structured proteins. So one kind of, you know, excuse to say is to basically say, well, that's all the model knows. It can only ever do structures, and that may be true. And we don't know yet. A kind of interesting hypothesis that I'm looking forward to testing is whether the model is learning sufficient amount of kind of fundamental concepts of protein sequence structure, et cetera, so that you can actually try and push it into these unstructured regimes. I don't know the answer. I wouldn't be surprised if you get what you learn and that's all you get, but it is an interesting question.
Thank you. Next talk. So with programmability, I think we can do an in silico protein evolution, so which can be used for tumor evolution study and vaccine development.
So, oh, sorry, I didn't let you finish the question.
As well as the virus and the vaccine development. Give some perspectives, comments.
Yeah, absolutely. So we can do, I think what you're saying is, couldn't we just do kind of in silico protein evolution? Why this fancy programmability layer? You can absolutely do trial and error, right? So you have to probably be able to evaluate billions of samples to kind of even think about optimizing things, which can get costly, right? You know, a minute of GPU time times a billion can still be a lot of money. So I think it does non-trivially improve efficiency if you're able to do conditional generation, because it means you can take far fewer shots on goal computationally. But I don't disagree that you can still use Monte Carlo sampling and other kind of techniques to explore sequence space.
Thanks.
Thank you for the great talk. So one of the advantages of the generators, as I understand, is that they can return sequences that maybe are outside the realm of directed evolution or naturally evolved sequences, you know, in the sequence space, right?
Right.
But most of the conditioning models that you might use are probably trained within those regions of the sequence space, right? So do you find that that's something that can be solved, where if you're conditioning on known sequences, does your generator typically return sequences that are similar to those known ones, or is there a way to keep the uniqueness of it while conditioning on known sequences?
I think it's a good general question. I would say it would all depend on the conditioner, right? So if the conditioner has limited extrapolation capability, so that, you know, it can really just kind of interpolate within the family that you learned, or whatever, you know, whatever the function may be, I think you would be limited to that. And so it's really thinking about, well, how can I build the best possible functional classifiers? You know, if you're thinking about something like enzyme design, right? And you can think about, you know, building some fairly generic classifiers of enzyme of catalysis that perhaps partially rely on, you know, some understanding of chemistry. On the other hand, if your conditioner says, you know, I want you to be an antibody, I've learned what an antibody is from a database of shapes, of antibody shapes, and I condition on that, then I think you should expect that you would get things that are shaped like natural antibodies. So I think, yeah, there's a lot of control we have over it, but it will ultimately depend on what the classifier is.
We have time for one more question. Please find Gevorg in the corridor after the talk. So go ahead.
Yeah, great talk. Yeah, so I have a question about, like, can you please elaborate on how does this model take cofactor or ions into account, and like if any of those 300 predicted had cofactor or ions in them?
Yeah, very good question. So this model and the one that's going to be published, it basically ignores all that, or I should say it strips all that information from the training set, and so therefore it can kind of implicitly understand that you can have zinc stabilized interactions or copper stabilized interactions or disulfide bonds, which it makes quite frequently. It doesn't know it makes them, but like, you know, it does, but it doesn't explicitly enable that annotation, and therefore you can't condition by the presence. But, you know, you can imagine it wouldn't be so hard to extend the model so that it can take cofactors into account. You can start conditioning on them and so on. But yeah, good question. Thank you.